Hello and welcome everyone. In this podcast we're going to start talking about transcription. And so in this first podcast we're going to just lay out down some of the basics of transcription. So let's go ahead and just write basics of transcription. And I want to begin with bringing back the central dogma. So remember, the central dogma said that we have a gene that is usually DNA, sometimes in some viruses it can be RNA, and then through the process of transcription, we can make RNA. And we'll talk in just a moment about some of these different kinds of RNAs that are produced. And then through the process of translation, we make protein. Today we're going to focus on this step here in this podcast. And the next couple podcasts we'll focus on transcription in eukaryotes and bacteria, how some of this modification occurs throughout the process. And then the next section, the next few podcasts after this series, we'll focus on translation. And then we'll bring it all together and talk about gene regulation. But that's several podcasts down the road. I'm going to talk a little bit right now about this idea of making the RNA. The central dogma says the end result is these proteins, but sometimes we know that different kinds of RNAs are made and they don't make proteins. That is the product. The RNA that makes protein would be the messenger RNA. And we're going to spend the majority of our time talking about messenger RNAs. But it's also important to remember that as genes are being transcribed into RNA, the other kinds of RNA we find in our cells also are transcribed from the same genes. So genes don't just make proteins. Sometimes their whole purpose is just to make RNAs, and those RNAs do the job. So we have messenger RNAs, and I'm going to just make a list over here of the ones you also need to know in addition to messenger RNA. Ribosomal RNA, we'll find later on when we talk about translation that these are very important in forming the ribosome and catalyzing the steps involved in translation. And then we'll see tRNAs are important also in translation, and they're transcribed as well. And some ones that you might not have heard of in the past would be like the microRNAs. These are important in gene regulation. And then the small nuclear RNAs. These are important in a step that we'll talk about in a couple podcasts when we're talking about modifying the RNA that's produced, the messenger RNA that's produced. And then this last one I want you to know are telomerase RNA. We didn't spend too much, we talked a lot about the RNA, but we didn't talk about what formed the structure at the end of the protein, at the end of the chromosomes that provided that protection and that stability. So we did talk about how we have the ends here, which are the telomeres, so I'm going to put a T here. But it's not just the DNA. We didn't really talk, as I said, too much about it, but there are several kinds of proteins and RNAs that form this kind of cap at the end of the telomere that provides that extra stability and protection. Now there are many, we're discovering all kinds of RNAs that our cells have, and they all have the very unique and incredibly important functions. But for the purpose of this class, just focus on these. If you go on and take a course like a molecular genetics course, I'm sure you'll talk much more about these other kinds of RNAs. Okay, I'm going to go ahead and erase what we see here, and then let's start talking about the basics of transcription. Let's begin by making a list of everything a cell needs to accomplish transcription. We need a template. And what we're going to see is that there are some similarities to replication when DNA is copied, but there are also some important differences as well. So just like replication, transcription uses a DNA template. However, I think it's important to note some differences of how the template is used. Remember, with replication, you took the two strands of DNA, and through semi-conservative replication, we made two new strands of DNA by using each half of this DNA molecule as a template. And also, actually I'm going to write the word, or my abbreviation for replication here, because I also want to point out some other differences. also with replication, the entire genome is replicated. Okay, now how is transcription different? Well, transcription is different in a few ways. So let's begin with the fact that when you are transcribing a gene, that the template is only one of the strands. This is my abbreviation for transcription, TXN. So if you see me use that, know I'm talking about transcription. Transcription only occurs on one of the strands. And so when the RNA is made, we only have a single strand because we're only using one half of this as a template. So that's a pretty big difference. Another difference is that only a portion of the genome is transcribed. And the reason for this is that we only transcribe genes. And in some organisms, particularly eukaryotes, most of the DNA doesn't code for a gene. So it would be incredibly wasteful to transcribe the whole genome. In addition, in multicellular organisms, you wouldn't want to transcribe the whole genome because you don't want every gene to be expressed. In the pancreas, you don't want liver genes to be expressed. You only want pancreatic genes to be expressed. So only a portion of these are expressed. In a little bit, we'll talk about the polarity, talk about 3' and 5', but this is just to give you a general idea. So that is one thing we need for transcription, a template. I'm going to erase all this down here real quick. The next thing we need is, we'll call this raw material, the building blocks of RNA. And for transcription, the building blocks, this raw material, is ribosomal nucleotrophosphates. Very similar to what we saw with DNA, where we use deoxyribose nucleotide triphosphates, but the ribose version is written very similar. You have your ribose sugar, and remember the difference between the ribose and deoxyribose is for RNA we use ribose and it has a hydroxyl group. We still have our 3' hydroxyl group, and we still have our 5' phosphate. We also have attached here to the first carbon the base. And in RNA, remember, that base can be guanine, cytosine, adenine, or uracil, not thymine. So this raw material is used to make the new RNA. The next material we need, or next thing we need for transcription, is the transcription, again my abbreviation is TXN, transcription apparatus. And essentially what I'm saying here is we need the proteins or the enzymes necessary for transcription. We'll see that these enzymes are very much different than what we see in replication. Some of them serve similar purposes, like we need enzymes necessary to pull the DNA apart, so we have helicase type enzymes, and we have enzymes like the polymerases that add the nucleotides, but they're different, right, because we need an enzyme that's going to bind RNTPs. The last thing I'm going to write here, and this isn't really a requirement, it's just a reminder. Just like DNA replication, the template is read 3' to 5', and the RNA is synthesized in a 5' to 3' direction. We still follow the rules to make it anti-parallel, and we still are going to follow Chargaff's rules. So the C's will bind to the G's as they're making new RNA, and the G's will bind to the C's, of course. And the adenines, instead of binding to the thymines in RNA, they will bind to the uracils. Just so as we saw with adenine-thymine base pairs, adenine and uracil gives us 3 base pairs, cytosine and guanine gives us 3 hydrogen bonds. Okay, I'm going to erase this real quick, and then we're going to start looking at the structure of the gene. Okay, remember, as we look at the DNA here, I'm going to just draw it as a block here. We still have our 5' end, and we still have our 3' end, and it's going to be anti-parallel, that never changes. 3' to 5' on the bottom. And let's make some genes. We're going to have gene A here, gene B, and then gene C. Remember, on the previous board I said that only one of the strands is used as a template, and it still follows the same rules of reading the DNA 3' to 5', and new RNA is made 5' to 3'. Now there is no rule that says the top strand or the bottom strand has to be used. Each gene may run in a different direction. Let me explain what I mean. Let's look at gene A here. Remember, the requirement is that it has to be read 3' to 5', and new RNA is made 5' to 3'. So gene A could be read this way, where the RNA that's formed from gene A here will be 5' to 3'. But it's going to be read on the bottom strand. Gene B may be read on the top strand, where you're going to read it up here. It's still reading, remember, 3' to 5', but the new DNA formed will be 5' to 3'. Same rules, just reading a different strand. And just to make things complicated, let's say this gene C is going to be read in this direction, the bottom strand, reading 3' to 5'. The new DNA is going to be 5' to 3'. One thing that doesn't change is that gene A, for instance, isn't going to read the bottom strand in one cell, and in another cell it's going to read the top strand. It's always going to read the bottom strand in this example. Same thing with gene B. It's never going to read the bottom strand, only the top strand. Now, as we know, there are always exceptions to the rules, and we're learning more and more about this every day. One of the conundrums with studying genetics is it's a relatively new science. It's just a little over 100 years old. And because of that, we are constantly learning new things. And so you may find out later on that what I just told you isn't exactly true. But for the purpose of this class, that's what we're going to stick with. That each gene can be read either in the top or bottom, but once that decision is made, so to speak, it never starts reading the other strand. Now let's take one of these genes and talk about the structure of the gene. Let's focus on gene A. So here's gene A, and remember, for simplicity, I'm just drawing it as two lines, but you should recognize that these are a double helix because it is DNA. And remember, this was 5' to 3', and then 3' to 5', and we're going to read the strand going this way, so it's going to be read this way. But how does it know to read this gene, to transcribe this gene? And we're going to talk a lot more about this structure here, but there's a structure called the promoter. The different kinds of proteins and enzymes are going to bind to this promoter to say we want to transcribe this gene. Down here is a sequence called the terminator. This will tell the enzymes that are making the RNA where to stop. Because remember, we don't transcribe the whole genome. We have to know this. The cells have to know when to stop transcription, and that's the job of the terminator. This part in between the promoter and terminator, we're going to call that the open reading frame. This is often abbreviated as simply ORF, for open reading frame. This is the part that codes for the protein in the case of making messenger RNA. And again, that we're going to focus mainly on messenger RNA today. In addition, you will have some regions here in some genes. Not all genes have this, but some do. And this we'll call regulatory sequences. We see these in both bacterial and eukaryotic genes, and they help add additional forms of regulation beyond just the promoter. When we in the next two podcasts, we're going to talk about bacterial transcription specifically, and then eukaryotic transcription. When we talk about these, we're going to talk about them in three different steps. These steps are initiation. None of these steps have a really complex name. So initiation. During initiation, you'll learn various enzymes and proteins will bind to this promoter to begin the process of making RNA. When they do this, they'll do similarly as we saw with DNA polymerase. It'll open up the DNA a little bit so that we can begin to break some of these hydrogen bonds to make the RNA. Then we'll have elongation. Elongation is always the simplest to explain, because really what's happening here is that the enzyme RNA polymerase will just move along the ORF, the open reading frame, making the messenger RNA, or other RNAs, as the case may be. And then finally we'll have termination. During termination, you get to the point where you said, we've already transcribed the entire open reading frame, now we need to stop transcribing, because it's not very efficient to keep transcribing these very long stretches of non-coding DNA. So the enzymes need to have a mechanism to stop transcription, and that's the role of termination. All right, that ends this podcast. Let's provide a quick summary. We went over the central dogma again to kind of highlight where transcription falls in there and what we're going to be doing in the next several lessons. We talked about the different kinds of RNA and how in some cases that RNA is the final product of the central dogma. It doesn't go on to make a protein. But with messenger RNA, it does go on to make a protein. Then we talked about we'll call them requirements of transcription. And then we looked at the basic gene structure. And within the discussion of the basic gene structure, we talked about which strand of the DNA is read within a series of several genes. We also talked about specific parts of the gene, like the promoter and the terminator, and these regulatory sequences. And then finally we talked about these stages of transcription: initiation, elongation, and termination. Alright, that's all I have for this podcast. If you have any questions at all, please make sure you let me know, and I'll be happy to answer any questions you might have. I'll see you in class. Bye.